This video a supplemental material for our manuscript named Rapid Global Characterization of Human IgG1 under Oxidative Stresses. In the manuscript, we describe a method that monitors 56 modifications on 18 of the 20 amino acids, and we discovered total of 53 modification sites on the protein. We tracked chemical alterations on these sites across multiple oxidative stress conditions with speed and consistency. There are 3 stress conditions were performed. The monoclonal antibody was incubated with leachable metals for up to 72 hours. Antibody incubation with hydrogen peroxide for up to 132 minutes, and with combination of these 2 stressors for 5 hours. And we have the non-stressor negative control, which was sampled at beginning and the end time points of other conditions. After quenching, protein samples were independently digested by trypsin and chymotrypsin. Digests were scanned by both data-independent acquisition, DIA, and by data-dependent acquisition, DDA, with our high-resolution mass spectrometry. Results of these scans were imported to Skyline data processing templates for both tryptic and chymotryptic digests. Tutorials of Skyline can be viewed on the website skyline.ms. Users only need to manually check LCMS peak retention time and peak integration accuracy, which we will show a moment later. Peak areas reported by Skyline is imported into the Global Characterization Data Processing Site, a universal automated data processing application created to batch process data, plot modification trends for peptides, generate sortable and downloadable modification tables, and produce JMOL code for 3-dimensional structural models of the analyzed protein. The sample preparation, digestion, data acquisition, and Skyline processing template generation are detailed in our manuscript. This video is a tutorial for how to use the Skyline templates and the Global Characterization site to generate all these data automatically. The source code of Global Characterization site, Skyline templates, and all testing data from control and stress conditions are uploaded to my GitHub. The link can be found in the manuscript. User might click in the Automatic Data Processing file for details of the source code. In the README file, users could find a brief introduction to this project, source code running environment, several packages that needs to be installed before running the code, the Skyline templates, report templates, and testing data from all 4 incubation conditions are contained in the folder above. To view the 3-dimensional molecular model, user needs to download JMOL. You may pause the video and get the open source software from the link. The X-ray crystallography data of the protein of interest could be found on the Protein Data Bank website, which we will come back later. Let us scroll up. Again, if you do not know how to use Skyline, the Skyline website have well-organized tutorials for every MS functions. To proceed, user could download all the contents in a zip file. Let us follow the download tab for the content. Inside the folder, layouts of all contents are the same as the web page. Let us open the folder, Skyline Data Processing Template. There are processing templates for each of the incubation conditions described above. Let us open the folder, Metals plus Hydrogen Peroxide. There are 2 Skyline templates for tryptic or chymotryptic digest. We can open the tryptic template. The file immediately starts search and import raw data that is being imported. Let us cancel it. The template includes all modified and unmodified peptides that we discovered in the scouting experiments. With the help of the templates, we can consistently monitor every modification sites. We can go to File, Import, then click Results, and navigate our way to the results. User could batch process and search modifications of the entire stability raw data. Thanks to Skyline, the data import will take about 15 minutes, so let us open a Skyline file that is already imported. We can see mass transitions of each peptides, and the well-aligned precursor and product LCMS peaks. All transitions in the templates has been carefully selected. Please refer to the manuscript for our criteria on deselecting and mitigating false positive identifications. For each sampling time point, we have 3 DIA scans for each of the triplicate digestions, and 1 DDA scan for one of each triplicate digestion set. There are 4 scans at each time point. For every identified peptide, the data shows very low mass errors, consistent retention time throughout the batch processed file, and isotope.product scores above 0.95. User only needs to adjust and ensure the retention time and peak integrations are selected. To export peak area, go to File, Export, and then Report. We have a report template for convenient automatic processing. To add that template, go to Edit List, Import, and find the folder in the Downloads. Select Skyline Data Processing Template, and select Skyline Report Template. Click OK. A report template called PTM Quantification is added. Select Preview. Let us enlarge the window. Totally process and plot all these modification data, and make the 3D molecular model takes at least 2 months if doing it manually. Here is where the global characterization site is needed. To import all the data to the site, click Export. Let us just save the file on the desktop. Name the file temporarily as trypsin digestion. Let us save it. Let us go to the desktop and open the file. We can scroll down and get all the data we need to process for global characterization. Remember, we have another chymotryptic digestion file containing similar amount of transitions. Before we import to the site, please do us a little favor. The sample name could vary drastically. Let us change the sample names to the sampling time points, 132, as 132 minutes. There is 4 scans per data points. Let us do it for the next 3 columns, and the same thing for the rest of the file. We will not bore you by typing this, because we have already done it and these files could be found in the downloaded folder. If you have a secondary digestion, for our case it is chymotrypsin, you may go through the same process and prepare the data for automatic data processing. Now it is time to crunch all these data. Go to our source code. We are running it on Jupyter Notebook. The code runs on every web browser except Microsoft Edge. We can drop in or select the primary digestion file. We can select either digestion. This time, let us choose trypsin as primary digestion file. Most characterization experiments usually use one digestive enzyme. In that case, just leave the second drop box and proceed. We used 2, why not drop in the second one? Let us select the chymotryptic file. For the protein subunits, let us select both heavy and light chains of the antibody, and do the same thing for the secondary file. We select cysteine carbamidomethylation as constant modification. If you have multiple constant modifications, feel free to select all. Notice that the site is not limited to any protease or combinations of protease, nor give any limits to the number of subunits or number of modifications. If users were to process modification data of eukaryotic ribosomal complex, all or any number of the 82 subunits could be selected. Thus, we say this is a universally applicable program for modification data processing in the manuscript. Now we can start plotting trends of modifications. Let us select Show Peptides with Modifications. Let us select a peptide that is shown in one of the manuscript figure. For the interactive part, we can change the X and Y axis scales. We can change the name of X axis. Let us just add minutes as unit after the incubation time. We can drag on each of the axes for a convenient view, or zoom in if some modification changes are too small, or zoom back. The dots and error bars represent means and standard deviations. Also, the site does not limit number of replicates. Peptides shown in the first plot are the ones fits our peptide selection rule. Let us go back to the slides for explanation. Since the site also works for any number of miscleavages, there could be multiple peptides covering the same protein sequence. Our code maximizes sequence coverage while give preferences to peptides with less miscleavages. Peptides that are deselected from the rule are shown in the second plot. There could be sequence gaps where the primary digestion is not covered. The program will pull peptides from the secondary digestion file, if there is any, and shown in the third plot. Most peptides in the gap area were not modified in our case. The site can generate a summary table for all the modifications. Let us select Yes in the drop-down manual. The table generation takes a little time. The table gives information on peptide starting and ending positions, peptide sequences, all modifications found on that peptide and their corresponding locations, and percentage modification mean and standard deviations at each sampling time point. The table can be sorted by which amino acid is modified. Let us select M for methionine. The table will show all the peptides that contains methionine modifications. It also takes a little time. We can see 5 peptides that contains modified methionine, that is all the methionine on the tested antibody. We can also type in a file name and select Yes on the drop-down manual to download the current table to a local disk. For the sake of time, we will not show here. Despite, it takes 5 minutes to format the table into a publishable grade and saves user lots of time. And this table is the one we used in the manuscript. Now let us generate the JMOL code for the 3D modification model. Since there is no intact X-ray crystallography data available for the protein we analyzed, we need to choose an analogous protein. The protein we chose in the Protein Data Bank or PDB is coded 1HCH, which is an intact human anti-HIV antibody. Let us download the PDB file first. Then let us navigate to annotations and scroll down to gene product annotating. Notice that the 2 heavy chains in the PDB file is coded as H and K, and light chains are L and M. We will need this information. Let us go back to the global characterization site. In the blank, let S input the PDB name for each chain separated by semicolon. So, it is HK for heavy chain, semicolon, LM for light chain. The next step is optional. Since our protein of interest and 1-HCH are different proteins, sequence alignment are needed for a perfect 3D model. I currently coded for specific alignment to protein 1-HCH. A universal alignment for protein of any numbers of subunits will be coded in the next version. However, this alignment is totally optional and will not affect functionality of this website for any other proteins. Now let us select yes to generate the JMOL code. After a short waiting, we have the code for all the sampling time points. Let us go to the last time point and copy all the code. You should have previously downloaded JMOL software already. Let us open it and drag in the 1-HCH PDB file into JMOL. Click on File and then Console. Let us paste the code in here. Voila, you have the global view of the 3D modification model of your analyzed protein. Exciting. That's all for this short tutorial video. Thank you for your interest in our manuscript and hope our work can be useful for you. If you have any questions, please feel free to contact the author. Thank you for watching.